So as we know, DNA is the instructions for us to build traits, and traits are made of proteins that are made inside of every single cell. So here's, for example, DNA instructions. DNA gives you the instructions to make mRNA. mRNA then gives the ribosomes instructions to be able to know how to connect amino acids together. So we're going to go through 3 common mutations that could occur inside of your cell. So first one is a missense mutation. So in a missense mutation, all that happens is that just one nucleotide changes, and that can change one amino acid. So for example, here's the DNA. This is only one letter, the G, has been changed. One letter got changed. It got substituted. Over here, the mRNA is made from this DNA instructions. And if you look at the amino acid table, right? So before you have CCC, right? CCC becomes GGG for the mRNA, and GGG for the mRNA would have been GLY, right? But over here, because of this one nucleotide difference, instead of GGG, right, you have CGG. And what's CGG? CGG makes ARG. So the amino acid became different now. Only one amino acid. That's a missense mutation. Now you might be wondering, is it possible to change one nucleotide and not change the amino acid? Yes, that is possible because, for example, let's look over here, the CCC over here, let's pretend we change the G to G at the end. So CCG and then GGC. So over here, we look over here, GGC is GLY, and it's no different than if it was GGG. Okay, so sometimes one nucleotide change can make no difference in the final amino acid that you make. But other times, as you can see, it can change one amino acid, and that's called a missense mutation. So here's an example. Let's say you have the instruction, go to the store and get chocolate milk. A missense mutation would be, go to the store and bet chocolate milk. So you can see just one letter got changed. The next example of a mutation would be a nonsense mutation. So in a nonsense mutation, one nucleotide changed, but as a result, you get an early stop codon. Now, as you remember, stop on the amino acid table, it tells you that you need to stop making the protein. You need to stop doing translation. And so when the ribosome sees the stop, it's not going to connect more amino acids. And so here's an example. I made up a new original DNA. So DNA is this, you get this RNA, and when you get this RNA, right, you can see here, right, AUG is MET, and then CAA, right, so CAA is GLN, and then UAA is stop. So I don't write anything else because the ribosome stops. It doesn't use this instruction anymore. Now let's look at the mutation, the changed DNA. As you can see, the changed DNA has just one letter different, one nucleotide different. Now the RNA has just one nucleotide different, but now you can see instead of building the second amino acid, it just makes MET, and the second instruction, UAA is already stopped, so then we don't get any more amino acids. So a nonsense mutation, it causes the protein to stop early. And so here's an example of that. Go to the. As you can see, depending on where the nonsense mutation is, it can make the protein not function correctly, because if I say go to the store and get chocolate milk, that's completely different than saying go to the. You don't know what I'm going to say. Go to the what? Go to the park, go to the store, go to where, right? And so the same thing happens inside of your body. The final one, frameshift mutation, is probably one of the worst mutations because you're going to see how all the instructions get changed. So here's the original DNA. And in a frameshift mutation, it causes a new nucleotide to be added or removed. So in this situation, I'm going to pretend I add a T at the very beginning here. And as you know, every 3 nucleotides allow you to have instructions to then produce an amino acid. And so here you can see from TAC, CCC, ATT, right? That was the original DNA. And we said that this original DNA, right, was able to make MET and GLY, right? So now let's see what happens. Let's pretend the mutation added the T to the front. As you can see, everything got shifted, right? So the TAC got moved over. Here's the TAC from before. Here's the CCC from before, CCC, and here's the AT, right? And this last T is not even counted because just one T by itself is not enough instructions. So here you have TTA, which becomes AAU, and instead of getting MET first, now AAU gets you ASN. So here's AAU, ASN. Okay, luckily here, the second amino acid is the same, but now there's a third amino acid that didn't exist before, because before we just told to stop, but now we actually have instructions to make a different amino acid. So GUA is VAL. Okay, and so that's how mutations affect you. When they change the DNA instructions, you also change the RNA instructions, and when we change the RNA instructions, different amino acids get put together. Just like in this example here, a frameshift mutation can be like chocolate store and many cat poop. So you could see the instructions could have gotten scrambled, and then new instructions could have been created because of the way that the nucleotides moved. That's it.